let me finally look at galactose metabolism. So of course we get galactose majorly from milk, that is lactose. Lactose is a disaccharide that is made up of glucose and galactose. Okay. And galactose can enter glycolysis by it being converted to glucose 1-phosphate. And this is happening in the liver. Although galactose is not a very important, it's not a very important monosaccharide or other sugar for energy production, but it can be converted to glucose 1-phosphate and get into the glycolytic pathway or even the gluconeogenic pathway. So how does it then end up becoming glucose 1-phosphate that can get into glycolytic pathway? So what happens is that galactose is phosphorylated by galactokinase to generate what we call galactose 1-phosphate. Now, galactose and glucose in their structures, they look more or less the same. We've seen the cyclic structure of glucose. So it's more or less similar to galactose. Of course, there are changes at carbon number 3, 5, and 4. Yeah. So, which means carbon number 6 of galactose is being phosphorylated with the help of galactokinase. Then epimerization occurs of galactose 1-phosphate to glucose 1-phosphate. So we are converting galactose 1-phosphate to glucose 1-phosphate. And I said these structures are related, glucose and galactose, and that is why it is just epimerization. It's more or less like an isomer of the other. Now, for this epimerization to occur, you require UDP. You require UDP uridine diphosphate. Okay, so this uridine diphosphate is obtained from uridine diphosphoglucose or UDP glucose. So it's uridine diphosphate attached to glucose. So that is where epimerization can occur. So you get it from uridine diphosphoglucose, so uridine attached to glucose, and you require an enzyme called galactose 1-phosphate uridyl transferase. So it's going to transfer the phosphate from galactose to glucose at position number 1. Okay, so when that happens, then you're going to get two products. You're going to get, initially you had UDP joined to glucose. So epimerization is occurring. So you are ultimately going to get uridine, UDP joined to galactose, and glucose, which was originally attached to the UDP, will be phosphorylated at position 1. So 1-phosphate and UDP galactose. That is what is happening. So precisely this is it. You have galactose from milk. With galactokinase, you get galactose 1-phosphate. And of course, with the use of UDP diphosphoglucose and the enzyme galactose 1-phosphate uridyl transferase, you are able to generate glucose 1-phosphate as UDP galactose. And remember, you need UDP glucose for this reaction to occur. Now, once you have glucose 1-phosphate, we know that with phosphoglucomutase, you can be able to generate glucose 6-phosphate. And we know that glucose 6-phosphate is an intermediate in the glycolytic pathway. Once you have that, UDP galactose is epimerized to UDP glucose by UDP galactose 4 epimerase. Okay. So UDP portion is exchanged for the phosphate, generating glucose 1,6 phosphate. Then, of course, glucose 1,6 phosphate is phosphate using. After this class, I'm going to share with you notes for glycogenolysis. Of course, fructose and galactose metabolism, hexose monophosphate pathway, and disorders of carbohydrate metabolism. With that, I'll believe that we've comprehensively covered what we call carbohydrate metabolism, and we'll move to the next step, I think, which will be lipid metabolism in the next class. So I think that is okay. And if you're encountering any hardships in the notes, especially for those that have not covered the hexose monophosphate pathway and diseases or disorders of carbohydrate metabolism, you know how to get me. You can always consult, you can always ask questions. I'll be ready and willing to answer. Yeah, so do want to welcome any questions concerning today's class, either of these three pathways, any elaborations would want made. And if there are no questions, then what we'll do is call the class to an end. Then I'll share the notes with your class reps.